All right, so today I will talk about mechanobiology in neuronal development. Most of the things that I will talk to you today is, or a big part of it, is from basic textbook on neuroscience. And the idea is first give an overview of what neurons are, how do they work, what is their structure, and then if I get time, I will get into a little bit of some of the research that we are doing. So the idea is some of you are from engineering, some of you from biology, some of you are from physics or material science. Thanks. Yeah, great. Thanks. So in case this lecture appears to be repetitive, my apology, but I'm sure some of you may benefit from this one. But the main emphasis here is to have a discussion about neurons in general. So I have a lot of quizzes on the way. So get ready. All right. So the outline is the basics of neurons, structures and functions, early development and growth, and role of mechanical forces on neurons. So I have lots of slides. I think I have a couple of hundred of them. So I don't expect to finish. I can finish whenever my time is up. But I want you to participate and ask questions. And I may not be able to answer many of your questions, but at least let's have the questions. Okay. Quiz number 1. How long is a neuron cell? Yeah, raise your voice.
All the way up to a meter.
Could be a meter, okay. Does it surprise some of the engineers here? No, it's very long. So my neurons could be starting from my central nervous system all the way to the tip of my finger, right? This is very, very long, pretty unusual. Second question. So neurons are very, very long. This is an important information to keep in mind because that's where the mechanics will show up towards the end. How long do they live? Depends on time or type.
Could be long life or whole life.
As long as you are. Okay, good. So the neurons don't divide. Once they form, that's it. My grandmom's neurons are as old as she is. Unlike many other cells that regenerate, divide, and keep on continuously growing. This will be another part that will show up towards the end through the mechanics. What do they do? This is a more general question. Give me 2 answers. What's that?
Transfer signals.
Transfer signals. Very good. Transfer signals. That's one of the primary jobs, right? And a basic, simple job. So the moment I'm trying to decide to move my arm, my brain sends a signal to the right neuron that contracts this muscle by sending a signal to the muscle. How long does it take? Do I first think that I'm going to move my arm and then send the signal and then show up my arm is moving? Or instantaneous? I don't even know when did I decide and send the command. Give me an approximate time.
0.2 seconds.
What's that?
0.2 of a second.
0.2 second? Shorter than that.
0.01.
It's on the order of a millisecond, maybe a couple of milliseconds. Okay? It's so fast. So remember, these are long, they live long, they act very fast, right? So all of this will show up. And I'm hoping by the end of the lecture, you'll have some understanding of how they achieve all 3. Okay. So I'll have this picture over and over again. Okay? By the end of the talk, you'll be bored with this picture. But this picture is significant because this is an artistic rendition of a neuron. They are very, very long. So they form connections with one another. So if I think of this as coming from a cell of a neuron, this cell would be probably going all the way to the next building, to the next city. Yeah, and then it forms these kind of connections. And how many cells are there? How many neurons are there in the brain, in our brain? Throw some number.
Millions.
About 100 billion. And 100 billion cells probably making trillions of junctions, right? Many junctions. Okay. So starting with the basic morphology of a neuron. So there are essentially 4 major parts of any neuron cell, or most neuron cells, not any, most neuron cells. And so these are the major parts. The one part or one segment of the neuron are the dendrites. There could be very, very dense network. Then comes the cell body with the nucleus inside. And then very long cable, we call it axon. And towards the end of the axon, there are little branches that could form connections with the other neurons or other muscle cells, primarily. So the part of the dendrite is where the signal is being, in general, is received. If somebody stretches or light comes in or voltage comes in, this is the region where the signal first is, where this part is stimulated first. And then the signal goes through this very long axon. So the job of the axon is to conduct the signal on the order of millimeters to meters, depending on which animal. And the job of the synapse, where it forms a junction with the other cells, muscle cells or neurons, is to transmit that signal to another cell. Okay? So somebody mentioned that their job is to transmit the signal, take signal from one side to the other. Now, if you ponder a few seconds, then many questions pop up in your mind right away. So if you think of this guy, is on the order, for me, for you, it's on the order of maybe a couple of feet long. Things have to happen in about a couple of milliseconds. The moment a mosquito bites my hand, snaps on it, right? So it has to happen very, very quickly. So what are the processes that have to happen? How does it get so long compared to when I was a little boy or a baby? What are the essential components that has to happen? In the axon, even though it's a long cable, there's one thing that I should mention before I forget, is there are regions which are insulated by what we call myelin sheets, and there are little openings in between that have a specific function. And so that's, in general, the overall structure of the neuron. The body of the cell, the cell body, it processes most of the proteins and the protein generators, the mRNAs that are distributed all along the length of the axon, and to conduct the signal. So if you now look at when the cell body are producing these proteins are packed in small vesicles. They have many names. Some of them are larger, which are called core dense vesicles. Some of them are smaller. But the bottom line is, this is the factory. The cell body is the factory that is producing a lot of proteins. And many of these proteins are transferred along this long axon all over until the synapse is reached. Some of them are packed in small vesicles, and also they're transferred all the way toward the synapse. Again, keep in mind, these are long, right? The diameter of this axon varies from at different stages of development. At the early stages, of course, small. By the time we are adult, they're on the order of tens of microns, but small. So if you think of something that is on an order of 10 microns long, 10 microns in diameter, about half a meter long, you are talking about orders of magnitude, right? A small cable that goes huge distances. So the challenge then is, how do you transmit these chemicals that are being produced in the cell body in these little vesicles that has to come all the way, or other which are not packed in the vesicles have to be transferred all the way. So that's one of the job of these neuron cells. They're not only transmitting information, which would be an electrical information, but also transmitting these chemical species that are going along the axon. At the synapse, as we go along down the length of the axon at the synapse, again in artistic rendition, these vesicles are clustered. These are called neurotransmitter vesicles. They are carrying neurotransmitters. They are almost the heart of all the neurons. These neurotransmitters have to be released to the muscle cell or the other neuron and the job of this neuron is done. So a signal comes, that signal becomes an electrical signal that has to run all the way on the order of a meter, all the way coming to this end, and at this point, these neurotransmitter vesicles are released to the next cell, and the information is passed on to the next one. Okay? So these are called synaptic vesicles. And the way these synaptic vesicles work is when this voltage signal comes, these vesicles that are clustered at the end has to release their content. So they have to go through exocytosis. They merge with the membrane of this axon, or of this membrane of the axon here, and releases their content just by fusing with this membrane. And these neurotransmitters, there are quite a few of them, these neurotransmitters are released, and these neurotransmitters then open up the ion channels or the gates to the next cell. If this is a muscle cell, then these gates open and ions get in and the muscles get contracted. So very briefly, the moment I decide to walk, these neurotransmitters have to be released. My muscle gets these neurotransmitters so that their ion channels can open. Ions get into the muscle cells, and that makes the muscle cells contract, and I am coordinating my movement almost without knowing what's going on. So thousands of signals are going through this neuron's movement at a millisecond time scale, extremely coordinated way, and I'm walking without any thought process going on behind it, right? So again, the only time-limiting step, or one of the time-limiting steps, but a major one, is the diffusion of these neurotransmitters from this, what we call presynaptic terminal to the postsynaptic terminal. It's about on the order of 100 nanometers scale, and this process relies on diffusion. When they're released, they diffuse to the next cell, and this takes about 2 milliseconds. This is one of the primary limiting steps in the process. So what would happen if these neurotransmitters were not in the vesicles? If nature would produce them in the cell, and they would be distributed all over the axon, and when the action potential comes, they would be released by just creating little holes. What would happen? Why nature chose these vesicles?
Control the concentration of release.
What's that?
The concentration of calcium.
Right.
You pack it into one package.
Yeah. So the content are inside are the neurotransmitters, so they would not be concentrated anymore, right? Diffusion would be always working, and they could not be packed here. They'll be diffusing everywhere. So the amount of release, had there been a process like that, the release would have much less in concentration. So it would not be very efficient. Okay? And when it is released, this is more close-up view. As the release process goes on, these contents come out, and some of these vesicles that are released are also endocytosed. So they go back and they get refilled, and they could be recycled. So this is called kiss and run mechanism, where some of the vesicles could be used. If this process were not there for endocytosis, what would happen? If you would have these vesicles come out and add to the membrane of this presynaptic terminal, if this endocytosis was not there, what do you expect from an engineering point of view? What's that? The membrane would just become indefinitely long, right? Because you're adding little membrane all the time, right? So it has to have some way to recapture some of the membrane area. That's one of the processes. So evolution has ended up having a process where this recycling typically goes on for about 5 times, and then some of the vesicles are being transported back all the way for degradation. Okay? So it's a continuous process. Okay, so now that you know the vesicles are produced in the cell body, They have to be transmitted all the way to the synapse. How long would you expect for a vesicle to reach from, let's say, from the cell body in the central nervous system to the tip of your finger? Give and take 2 feet long, diameter of 10 microns. So what are the processes that you could think of for this transport? Are you from biology? Bioengineering. Shh. Okay, engineers. You have heard already. So we could think of that cells are producing these vesicles, and they're producing a lot of them. So when you put a lot of sugar in one place, what happens? Sugar gets diffused, right? We call it diffusive process. So the cell could do that. It could produce a lot of vesicles. And because of this lots of vesicle concentration, they would diffuse anyways, everywhere. Okay? If that's the case, then one can imagine how long would it take for the vesicles to reach all the way to the end? Okay? So what theory would you use to calculate that, to estimate this? Just based on diffusion.
Fixed law?
Yes, fixed law and some diffusive constant, right? So you can think of this little vesicle that is being somewhere. There's just or here. Oh, right there. Okay. Yeah, thanks. So you can imagine that there's one vesicle in a fluid, and if you put it on the fluid, then the fluid particles bombard the vesicle, and the vesicle would start to move. And eventually it would sometimes go over here, sometimes go over here, sometimes go over here, and we call it diffusive dynamics, right? And the diffusivity of this particle, d, is given by something like 6 pi eta radius of the particle, and on top is this kBT, the Boltzmann constant absolute temperature. What is this equation called? So R is the radius, eta is the viscosity, absolute viscosity. So you can imagine for room temperature, body temperature, and vesicle diameter of maybe about 100 nanometers, and assuming viscosity of water, you can get an estimate of how long would it take for the vesicle to start moving all the way. And the root mean square displacement is approximately equals to dt. This is time. So eventually these particles would reach based on this concentration gradient. And if you do this, if you put the numbers, you would find that the particles, if it was only left to diffusion, would move on the order of about 1 millimeter a day. Okay? Approximately. That wouldn't do the job, right? If it takes a millimeter a day, that wouldn't do it. So what nature has done is it is not relying on diffusion anymore. This is the calculations which I just went through if you put the numbers. So what nature has done is it is producing some motor proteins. So these are the tracks that are running down this long axon. So the motor protein, there are several motor proteins. Let's say one of them being kinesin motor proteins. They hold this vesicle on one side and keep walking along the microtubules, which are one of the cytoskeletal component, along the axon. So they act as like tracks, and they walk pretty fast. Okay? So how fast do they walk? Here's a video. So if you look at this vesicle trafficking here, can you see it from distance? Vesicles are moving back and forth. Can you see from the back? Okay. In fact, we took this video last week from a fruit fly embryo. So it was an embryo that just formed the neuromuscular junction, which I will get to in a minute. So these vesicles are moving on an average of about half a micron to 1.5 microns per second. So if you think of the vesicle dynamics driven by the molecular motors, the speed ends up being on the order of velocity becomes, so if you add the motors, it becomes on the order of 400 to 500 millimeters a day. So 2 to 3 orders of magnitude higher than what your diffusion would give you. It is surprising that if you use these simple calculations and these simple motor dynamics, you get these numbers more or less to the point. Okay? So if you had diffusion, it would take long time. So motor proteins are used to move these vesicles back and forth. Okay? Sometime in the early 50s, this was a big question, that how does axons transmit, these vesicles transmit this information? So one experiment was done, which is putting radioisotopes in the cell body and seeing where they are. And it turned out that it was found out that they move much faster. It turned out later on that axon uses both of the mechanisms, both diffusion as well as this motor-driven dynamics, to transmit the information from one place to the other. One is called the slow transport, one is the fast transport. So the fast transport is done for these vesicles because they are larger. When we talk about 100 nanometers, they are large, which makes their diffusivity much less because this is the radius as the denominator. So it will take forever for the vesicles to go from one place to the other. No, that is the speed. So when I decide to move my arm, I don't have to have the vesicle starting from the cell body all the way to the end. Vesicle's already there. So what's the confusion?
Does that ever lead to an issue where you can like overexcite the neuron and not have enough vesicles?
Oh, okay. Good question. I don't know. Can you excite enough that you release everything, right?
Yeah.
And there's nothing left, right?
Is that
a disease?
Yeah, I was going to say, or drug induced with some.
Can you
recreational drugs that involve changing the postsynaptic terminal?
Yeah. So one example is when the snake bites the frog, right? So what does it do? So it releases the content that is being released. I think is acetylcholine. So the snake actually first bites the frog, and then paralyzes it, and then it happily goes and eats it. So what happens is the snake venom, I don't remember the name of the venom. So what it does is it comes all the way and it consumes the acetylcholine, which is the neurotransmitter. So even though the frog wants to jump, it is firing. Acetylcholine is coming out, but it is unable to activate the muscles. So yeah, it just keeps firing, and until its death, it keeps firing. I don't know of any case where you can excite such that you can release. I have to check that. Don't know the answer. Be interesting. Yeah.
Wouldn't that be? Wouldn't the action potential actually, like once it reaches a threshold?
Yeah.
Actually, the depolarization stops. So even if there is a lot of depolarizing signal, it doesn't really go away.
We'll get back to that one. Yeah. We'll get to that one. I'm trying to understand your question. So if it is moving at 400-500 millimeters, how come we move so quickly? Is that a confusion? Mm-hmm. Okay, so there are two things here. The second part, I'm glad that you raised it this way. The second part is, well, the vesicles are moving as fast as they can on the order of 400 to 500 millimeters a day with motor proteins. But there is another velocity involved. That is the action potential velocity, okay, electrical signal. So the vesicles are there waiting for the electrical signal to come, to be fired or to be released. Vesicles are moving at 400 to 500 millimeters a day and sort of marching towards this end. And once they march, they have enough soldiers waiting for the command. And then the action potential comes at a much, much faster rate. That is like turning on a light, and then immediately the light goes to the other end, right? When we talk about millisecond, it is that speed, not the speed of the vesicles. But speed of the vesicles is also important because unless you have that much speed, it's going to take forever to fill it up. Okay? Okay, so this is again a quiz because I want to make you think or reiterate what we have said so far. So how long will it take for a vesicle to reach your fingertip once it starts from the central nervous system? Well, given this speed, 400 to 500 millimeters a day, you might be on the order of half a meter, right? Just a couple of days, right? Or maybe 1 day to reach. So the cell is producing these vesicles, and now from now on you would imagine your cells are producing these vesicles. They're taking a day, you sleep tonight, and the next night they're there on an average. Okay. So we talked about transport and structure. Now what is the function of the neuron? So now we are going to get into the signal transmission as one of the primary functions. Okay, so this is a busy slide. By the way, again, I want to reiterate, most of the slides are coming from a single book, which is the Kendall's book on principles of neuroscience. Okay, so I don't want to put every slide the same title because I have put it at the beginning. So what happens? So this is a long axon. It is one end of the axon. It may be connected to something that sends a signal, and once that signal comes as an electrical impulse, and we'll get to it in a minute, it is usually a voltage spike. That voltage spike travels to the long axon, and by the time it comes to the other end, that electrical spike or the voltage spike causes these neurotransmitters to get released. If there are very few spikes, then few transmitters get released. If there are many spikes, many vesicles get released. Many spikes, more vesicles get released.
How is the electrical signal transmitted?
Good. That's coming next. Okay, so somebody asked, can you overstimulate continuously, right? So now that I'm thinking in this figure, so you have lots of spikes. So what we know is if there are a train of spike, then the vesicle density may go up in the presynaptic terminal. So let's say you start with 100 vesicles, and then your several pulses come and you release. What it turns out that if number of pulses is very few, then they would go back and restore to the original value, 100 vesicles being released, being ready to be released. But if there are series of pulses begin to come, and that keeps on coming, then the number of vesicles in the presynaptic terminal may also go up, which is like the presynaptic terminal has a memory that I've been used many times. Okay? So Eric Kendall, who got the Nobel Prize in 1992, gave the biochemical pathway by which this increased activation of the presynaptic terminal is achieved. So to answer this question that can you overuse, I don't know the answer, but what we know is that if a synapse is being used many times, which means many times the vesicles are being released, there is an inbuilt mechanism by which it not only replenishes what it had before, but it over-replenishes. So that next time the vesicles are released for the same action potential, it may release more. Okay? So that's a form of memory which we call synaptic plasticity. If we have time, we'll get back to it later on. Okay?
So the reverse is true. If you don't use the neuron, you will actually deplete.
In the early development, that's what happens. So you can what is known as the pruning. Right? Initially, when we're born, we have a lot more neuronal connections. The connections that are not being used begin to be pruned out. So as you progress during early development, you can see less and less dense is the network.
So some people have faster reflex, some people have slower. So does it have to do with like how fast vesicles actually transmit? Or
Some people can think faster. Is that what you're saying?
reflexes in terms of like movements or
Oh, motion.
response to some stimuli?
Oh, I don't know. I hate to say and give an answer. Okay, it's fairly complex. Unless somebody else knows the answer. How do we get more?
I think there are two different questions. Responding to a stimulus and active stimulus. I think active stimulus mainly in your muscle types. But responding to a stimulus, I think, I don't know.
Basically, how well you train yourself, right? So the athletes can start running as soon as he hears the firing of the gun. But my response would be less, right? Because I'm not trained to do that.
So it does not have to do with how well connected or how much vesicles get released.
It probably does. If you look at the literature of training, let's say the training of the mouse, for example, the more repetitively this is being done, the more vesicle, more efficient is the synapse. It can create more postsynaptic current for the same event, which is well established. So when the runner or the one who is competing in the Olympics gets ready and listens to the firing, he or she has gone through this process thousands of times. So the response time of this individual has been trained to be very short, right, by repetitive activity. So in that case, this person may have much more vesicle clustering at the end of the neurons that he needs to activate. Okay? So those are synapses that have been trained to activate much faster or much more efficiently. Okay? So the training is a big part of neuroscience. Okay, so now going back to the question that some of you have raised already, the action potential. This is a difficult topic to understand. What I did not mention in the previous slides is, well, what I mentioned before is the overall structure of the neuron. The neuron has this axon, has a cell body, the dendrites, and the synaptic and the synapses at the end. And this axon has these microtubules, which are the tracks for the vesicles to run down and back, right? And these vesicles have to be moved and being clustered at the end to be fired. What it did not show is what's happening along the axon, what's happening on the membrane of this axon or dendrites. It's very, very rich. How rich? The membrane of the axon or any other cell, what happens is on the membranes are extremely rich with many, many things, one of the components being the ion channels. Okay? So what are ion channels? Ion channels' main job is to allow ions to go back and forth. So ions can go through back and forth. There are some ion channels that are there that are passive. They let some ions to go through and some ions not to go through based on size, based on charge. There are ion channels which only act when there is a significant voltage difference between the two. So it's like a circuit where when a certain threshold voltage is exceeded, those ion channels will open. They're called voltage-gated ion channels. So example being sodium ion channels and potassium ion channels. Okay? So this membrane is very rich with these ion channels. They also have cholesterol and many other things, but ion channels being one of them. Now, if you look at the membrane, what happens is because of these ion channels, one side, the extracellular side of the ion channel, happens to have more positive charges just on the outside of the membrane. The inside has more negative charges just because of this ion channel's activities. So that creates an electric field. So electric field is going down, and typically this is on the order of minus 60 to minus 70 millivolts. So a membrane not being touched, undisturbed, has the intracellular contents here, the microtubules, the cytoplasm, and so on. On the outside is the extracellular matrix. And what you have is the membrane that is acting like a capacitor. It has more positive charges on one side than the other side, and you have an electric field in between the two. So everything being undisturbed, it stays there. Okay? What would happen if you start having a leakage somewhere over here, such that some ions can move? So for example, outside is a huge amount of sodium ions, about 450 millimolar concentration. Inside, sodium is very small, about 50 millimolar concentration. Inside, calcium is very, very high, about 400 millimolar. Outside is very, very small. So in terms of the total charges, outside and inside, it has to be charge neutral, except this one layer of charges, one or so layer of charges, just outside the perimeter, outside the cell membrane. But overall, you have a charge neutrality, because sodium ions are large in quantity outside, and potassium ions are large inside, but there are other negative ions which are balancing it. For example, the ions that are contributed by non-metallic components is very high. So inside, overall, you have a charge balance. But what you have is a large concentration of potassium ion inside and large concentration of sodium ion outside. So if I had a pore that is being opened up, what would happen? This is very important. I want to make sure that all of you get this idea. Without this, action potential doesn't make sense. So there's an electric field, positive to negative. So if I put a positive charge outside, if you let it go, what will happen? So I have an electric field. I put a positive charge. What will happen to the charge? Just go run down to the negative side, right? So there's an electric driving force, right? But is there any other driving force? going back to the concept of diffusion. Yes, and what is this called? It's a concentration gradient, right? So the sodium, being high in amount outside compared to the inside, has a concentration gradient that's going inward, right? So if I let a sodium ion free to go into through an open channel, the sodium has the potential to go inside, right? Because of the concentration gradient. But it is also subjected to the electric field to go inside, right? The same way for any other of these ions. So as long as these ion pumps are working and maintaining this balance, you would end up having some of the ions wanting to go in, some wanting to go out, right? So it's kind of a floodgate that is kind of maintained, balanced. And when you need, you open up. When you need, you close. Is this clear? Okay, so now let's see what happens. So first of all, this ion, this membrane potential, or so this is called what is called polarized membrane, because it is positive charge outside, negative charge inside, right? So if this balance is disturbed, we would call depolarization, in which case you would put more negative charges inside. So now if it is minus 75 or minus 60, depolarization would mean that your electrical field is changed to, let's say, minus 30 instead of minus 60. So there will be depolarization. Repolarization would be going back to this negative value or more negative value. Okay, these are just the terms. So when I call depolarization, I mean that I'm allowing some charges to flow such that the electric field is imbalanced. Okay, repolarization would be becoming more negative. Hyperpolarization would become more than 60, maybe minus 100 millivolt. Okay. So I talked about ion channels, sodium channels and potassium channels. So what happens? So when there is a depolarization, so for some reason, for example, I stretch my arm, I shine a light, somehow I depolarize it by multiple means. Okay? So if I do depolarize, the sodium channels, where are sodium channels? The sodium channels, initially at rest condition, it is closed. Okay? It is closed. It's not allowing any sodium to go through. When I depolarize, then the conductance to the sodium channel is increasing. So if you think about the conductance of the sodium channel, it is increasing as my depolarization increases. So initially I am somewhere between minus 60 volt. As my volt increases, I increase the conductance of sodium channel. So it allows more and more charge to get in. Okay? Another property of these channels is when it opens up, it allows some charge to get in, and then it soon closes. So it's a fast acting process. It opens up, lets some sodium get in, and then closes. And it stays closed for some time. It cannot open anymore that easily. It remains closed. Okay? And it's fast. Opens up, lets some sodium gets in, closed. On the other hand, the potassium channels are a little bit slower. So they're also responding to this voltage depolarization, but they open up a little bit later and let the sodium ions to go out. Remember, the sodium was high out inside. Potassium was high inside, sodium was high outside. So if the sodium channels open, what will happen? High sodium outside, sodium will start to get in, right? And conductance increases because it's more depolarized, right? What happens then? So diffusion or electrical field all together acting now, right? So sodium gets in, and when it gets in, it depolarized. So when it depolarizes, its conductance it becomes even more. It allows more sodium to get in. So it's an unstable process. So once you start sodium channel open up, it keeps on opening up more and more. The conductance goes up indefinitely, right? Because the more depolarization happens, the more sodium gets in. When will it stop? It's balanced by electrical field, right? So if more and more sodium gets in, then inside would be now more positive than negative. So eventually, the inside voltage goes up compared to the outside, or electric field now changes its gradient. So inside becomes positive, and sodium is being positive, would have to overcome the positive charges. So even though outside high concentration, so it would eventually come to a balance. That balance is established when sodium reaches about positive 55 millivolt. It's not shown here, but it's somewhere over here. ENA is about 55. Okay? Only then the sodium will stop. Sodium channels will stop opening anymore. However, as sodium is getting in, the potassium ion channels are opening up as well. What are they doing? They are allowing the potassium to go out, right? To create, to reestablish the polarity again, right? When will they stop? Remember, sodium is high inside, right? So they are going against an electric field because positive is outside, right? So there are always two competing forces: the concentration forces, the concentration gradient, and the electric field forces, right? So at some point, the sodium will not go out anymore. That happens when remember, the outside is positive, inside is negative. Sodium is going against the electric field, right? So sodium will stop. Sodium will stop when enough sodium goes out until this balance is established again. So anyways, when sodium ion channels open because of the depolarization, because of some signal, because I saw an accident, because a mosquito bite, or somebody stretched me, somebody pinched me, somehow this depolarization started somewhere in one of these dendrites. The nearby sodium channels opened up, sodium starts to get in, and then potassium starts to get out, although there is a little bit of lazy potassium channels, so there's a little bit of delay. So eventually, the electric field looks like this. Initially, it was minus 55, but now I'm crossing, I'm going way up there. But sodium channels became tired, so they're all closed now, right? So by the time this imbalance happens, potassium was taking over, and this action potential happens, and it appears to be an electric field that changed. Okay? So it's like an electric conducting wire. You apply local electric pulse. What happens to the pulse? Well, somebody said it has to go away, right? Because inside, you have the cytoplasm with lots of ions, as you see in the previous picture. So you create an electric pulse that has to go away. So this pulse then starts to move downward fast until it reaches all the way to the end. Okay? So there are some important concepts here, which is that if I go back to the previous slide, maybe even before my first slide. Yeah, maybe this is fine. So all these processes are happening somewhere over here, and the electrical impulse coming down. But as the electrical impulse goes down, what happens? What do you expect? The amplitude of the impulse, what should happen as it runs down the decay, right? So the strength is decaying, right? So remember, I have to send the signal all the way to the end. So what happens for the neuron's case is there is a small region called hillock just after the cell body and before the beginning of the synapse, beginning of the axon. There's a region which is highly dense with sodium-potassium channels. So even though the signal is decaying, by the time it reaches at that region, because of this high concentration of sodium-potassium channels, what you get is a signal is decaying. Even though it's decayed here, at this point, there's an avalanche of sodium-potassium channel opening and closing. And you get a high amplification of the signal. Okay? So this signal goes way up. So when you take a cursory look on this action potential, you often hear the word threshold voltage. It's often used the word threshold voltage. When the threshold voltage is exceeded, only then sodium-potassium ion channels open and close. So it's not quite a fixed number for the threshold voltage. It is the point of instability. So when an action potential comes, when a signal comes, no matter what the value of the signal is, there is always a conductance for the sodium channel. It's not like a one value is either open or close. There's a conductance. It may be small, but there is a conductance. So what happens is there has to be enough sodium channels being opened up to get into the instability. Remember what was the instability? I put a little bit of sodium in, I depolarize, more sodium channels get opened up, I depolarize more. What is this called? Instability, right? You are falling from the mountain top. The more you drop, the higher is the velocity. That's called instability. But that instability doesn't trigger all the time. Because for low voltages, yes, some sodium channels are opening up. But at the same time, remember, there are ion pumps all the time. So the moment the ion pumps see that, oh, there's an imbalance going on, I have to keep the balance back. So those are not necessarily voltage-gated ion channels. They are always trying to keep it balanced. So when little voltage comes, little depolarization happens, yes, some sodium can get in, but it cannot trigger the instability. When the amount of sodium getting in is so high that it cannot be restored by other ion channels, then you cross the point of stability. So then it becomes unstable. Then it becomes an avalanche of sodium getting in because the other ion channels are not being able to balance it. Only other voltage-gated ion channels have to come in, which is the sodium ion channels. So that is the threshold. It's not a fixed number. Threshold comes from the basic concept of instability. So sodium ion channels are always conductive, but conductance increases with the increasing depolarization. This is a continuous function. Is this clear? Please ask if it is not.
Is this process the same throughout the animal?
Yes. Invertebrates and vertebrates both. So this study primarily was gained from the giant squid axon, but it has been shown for both vertebrate and invertebrates. Yes.
If you have an external electric field,
What's that? Yes.
will it actually affect?
You do the same thing. That's how neuroscientists do electrophysiology. You apply electric field, you put more current inside by probes, you create local depolarization, you create action potential, and you see the effect on the other end. Right. You can do it by light. You can do it by many other ways. Yeah.
So maybe that's how taser guns work.
What's that?
If taser,
Oh, taser.
yeah, does it work the same way by overloading our
Uh,
ion channel?
Taser is the torture equipment, right? Something like this.
It's a high voltage.
It's send a high voltage to the body.
It's done.
Oh, okay. Yeah, it creates, I guess, huge amount of electric field, and it create shaking, right? It is.
That directly goes to skeletal muscle, though. I don't think it.
Oh, but there are peripheral neurons everywhere, right? So there are sensory neurons, peripheral neurons everywhere. So you can always excite. But it's creating electric field to muscles as well. You touch it or you just shine the gun?
It's basically needles that stick in you and give you a, is it like 20,000 volts or something? Early large spike.
Okay, yeah, sounds fun.
Are there any specific spots on your body that's like more sensitive than others?
I don't know. All I know is that the typical knee-jerk response. So if you tap on it, your leg shakes. That's one of the regions scientists or neurologists check you. And if there's time, I can go through that as well. Okay, quiz. First one. What ion channels are primarily involved in generating action potentials? All of you should know now. Sodium, potassium ion channels. Second question, I haven't discussed that much, but you should be answering now. Because I intentionally left out one statement. Why does the action potential propagate both ways? Why not? If you really understood what I said, this should be obvious.
Sodium channels, when they close, it's too long time.
Right. The one that has been opened now, which is the wake, needs some time. So the only one that's available is forward. Very good. Is this clear? Anybody? It's not clear? So sodium ion channels which just opened need some time. Yes? Need some time to open up again. So if I have an action potential sitting here, you'd think of the choices both ways, going forward or backward. But it cannot, the one that had just opened a minute ago cannot open because it has a resting time. The one that is available is the one next. That's going to open first. Right? So it has a progressive movement downwards, not the frontward. Simple, right? It's simple because it's the time delay of the sodium ion channels to recuperate itself that makes it one dimension. It's an asymmetry in the system. The front one has not been activated compared to the back one. That's why it's forward motion. But the electric impulse would go both ways. If I have an electrical impulse in this channel, that could go both ways because that is passive. That is, if I put some charge would try to balance itself inside, right? But action potential cannot happen because, yes, the electric field will go both ways. But which ion channels are going to open? The one in front. So these action potential are produced. They are running down fast. But ultimate goal for them is to go to the end, right? The end is this synapse. And that's where this large density of these vesicles with neurotransmitter vesicles are waiting for these action potentials to come. So this action potential is nothing but a voltage pulse, right? So this voltage pulse comes all the way. And now the structure is a little bit different. So what happens is at this junction of the presynaptic terminal, it's loaded with a different type of ion channels. These are called calcium ion channels. So when this depolarization pulse comes, this large number of these calcium ion channels open, calcium ion gets in, and that facilitates the exocytosis process, and release happens. Okay? The job of transmission is done. So what have we learned so far is the vesicles have to be there clustered, because without this clustering, this process would be useless. Right? They have to be ready soldiers available, the action potential coming by the process that I described. Locally, this is rich with calcium ion channels. Calcium gets in, and a sequence of events have to happen, which we don't have time to discuss. But the bottom line is these vesicles then go through exocytosis, and the neurotransmitters get out and try to excite whatever is next. Okay? I wish I have. I will describe. Roger, what time do I have to finish?
Well, you've had discussion during the lecture, so
Yeah.
you have to finish by 10.
By 10. 20 minutes. Okay. If what I have described so far is very clear, then I can breeze through the next, which is the mechanics part. Okay? So what's happening during development? So the axon, think about yourself in the embryonic stage in your mother's womb. We are developing. These axons, these neurons are being produced. And what they have to do. At the same time, hands are being produced. Little babies, you see these pictures, so cute. You see their features, hands, brain, their features being developed in the womb. At that point, what's happening is these neurons, the stem cells which became neurons, are now growing, looking for their targets. What target? The neuron that forms the neuromuscular junction at the tip of my hand has to find its way towards the target, right? So in the very early development, these neurons are going long, long distances. We're talking about now hundreds of microns distances. When their diameter is very, very small, maybe on the order of hundreds of nanometers, are looking for their targets, traveling long distances compared to their dimension, compared to their diameters, right? So that's happening at the early developmental phase. So it's a big field by itself, how these axons find their way all the way to the end. It has to overcome all the temptations on the way. It finds all the muscles on the way. But it learned how to avoid those temptations to form neuromuscular junctions randomly. The one that is going to my fingertip avoids all the other temptations of forming neuromuscular junctions all the way until it finds the right target. It's still a mystery, I think, to me. It's a mystery. There are many models to describe which says, there are attractive forces, repulsive forces, chemoattractants, chemorepellents, until it goes to the end. I will skip this one. So, for example, this is an axon that is growing in a fruit fly, embryonic stage of the fruit fly, and what we are observing are the vesicles by shining light by fluorescence method. You might have seen this already, how you do the fluorescence imaging. So we are fluorescing the vesicles that are clustered, that are neurotransmitter vesicles. Okay? So fruit fly embryos, the fruit flies are the ones that come when the food is getting spoiled. You can see them around, very benign flies. And the embryo is about 0.5 millimeter in length. And here is the muscle that it would use to fly later on. This distance is on the order of 100 microns. The axon is about 300 nanometers, and it is progressing towards this target, towards this muscle. And we are highlighting the vesicles by fluorescing a protein on top, which are called synaptotagmin. Okay? When it forms the junction with the muscle, this density of the vesicles shoot up. They go significantly higher. This is common. Remember, vesicles at the synapse is much, much higher. Okay? So it formed the neuromuscular junction in about the 14th hour, and it has formed the target, and it forms the neuromuscular junction. Skip that. Just prior to this formation of the neuromuscular junction, the muscle is also waiting. So wonderful. The muscle that is knowing that the synapse, the target, is coming, begins to create these structures. They come out and try to handshake with the incoming neuron. Okay? So this handshaking happens in about 14th to 16th hour. And this is a video. I don't know how much you can see. This is the growing axon progressing towards this muscle. It's actually moving and trying to find its target. It's a very dynamic structure, everything happening in just a few minutes. And let's see. And here is the neuromuscular junction. This is the muscle, the green, the purple is the axon. And again, this is going back long distance. Okay. I think given the shortage of time, I'll skip this part. So life after synaptogenesis. So we form the neuromuscular junction or neuron-neuron junction as defined by the genetic code. Okay? So the brain is formed, neuromuscular junctions are formed in the early embryonic stage. Now comes the quiz again. How tall were you when you were on the 0th birthday? Take a guess. Few centimeters, right? Maybe 10 centimeters? 20 centimeters? 15 centimeters? 50 centimeters?
Wow.
Wow, that was tall. You were tall. 50 centimeters is half a meter, right? 35 centimeters? Okay. How tall are you now? What's the ratio? Five? 5 to 6. Think about a rubber band. The rubber band, let's say 5 centimeters long, and you want to make this rubber band become 25 centimeters. Can you do that? It's going to break, it's going to fall apart, something is going to happen, it's going to thin down quite a bit, right? So the point I'm trying to make is once these neuromuscular junctions are formed, neuron-neuron junctions are formed, the body is growing. The neuron doesn't divide. So what has to happen? The body is going to pull it out, right? The early part of growth was axonal, trying to find its target. Once the target is formed, this is under the mercy of the development of the rest of the body. The body is growing, the axon has to be stretched out. You talk about role of mechanics, that's essentially beginning of the role of mechanics. It had a little bit before, which I hope I'll cover a little bit. But then once the neuromuscular junctions, neuron-neuron junctions are formed, the body is growing up. Only 5% of the total growth was done during the growth process. And the rest 95% is done by body stretching it out, which is called as tau growth. How is this possible? Okay, so in case of a fruit fly, if you are a fruit fly, it's about 150 microns by the time it grows. It's about several millimeters. So these neurons has to be stretched by 5 to 10 times. Okay? So if you think of it as a material, this axon, which does not divide, to grow, and if you now think of what we discussed before, that it is loaded with ion channels, it is loaded with microtubules, the cytoskeletal components, all the vesicles, right? They have to grow all the way by 5 to 10 times. So what does have to happen? Can it afford to become thin down as it is being stretched? Because the transport will drop significantly, right? So what has to happen? What's that? So it has to grow by developing. It has to grow by bringing more cytoskeleton from the cell body. It has to bring more cell membrane to make the diameter bigger, right? Because the transport has to happen more as the development grows. So what could be the role of mechanics here? Tension-induced growth. Very good. Very good. So if you think just very simply, the one that just formed the neuromuscular junction, the axon has no reason to grow anymore because it found its target, right? It can perform. But the muscle is moving away from it. So the muscle is applying some tension on it. So the only way it has to keep up with the growth of the muscle is by growing. But it has to grow if there isn't enough tension. Think about this threshold again. So if a threshold is exceeded, the threshold of force is exceeded, maybe some signals come, and then more vesicles come, the more cytoskeleton comes. But that more has to stop if the tension is below a certain level, because otherwise it will be over-fattening up. So now you can think of like two thresholds, an upper threshold and a lower threshold. If the upper threshold is exceeded, then it begins to grow. More material is being transported. It's like the more trucks come to lay the next road so that more trucks can come, if the tension is exceeded. If the tension is below a certain level, there's no reason to grow. Right? Can the tension become zero then?
You lose the arm.
Oh, that's if you injure it, right? But I'm talking about if everything is intact. Just from an argument. If tension is a signal to these neurons to grow, then can the neurons afford to make the tension zero? No, because tension is the means by which it is either growing or not growing. So it has to maintain some tension then, somehow. Right? Is it clear? From a purely argumentative point of view. So this neuron, you remember the first quiz was how long are the neurons? You all said they're meters long. You all know how long you were when you were born. By the time you are now, you have grown by 5 times. So something had to happen to these neurons to grow that much. And the only way it could keep up with it is by growing. And by argument, we are thinking that it is probably the tension which does the job. And if it is the tension, then if the tension is a sensory signal for the growth mechanism to come into play, the tension cannot be too low. If it is below a certain level, then it doesn't grow, but it maintains the tension. So it waits for the tension to go up again. Okay? Beautiful question. If the tension is too low, the only way it can build up the tension is by shrinking. Right? Because if there is a threshold tension it wants to maintain, then the only way it can happen is it can shrink and build up the tension. Okay? Is this clear? I haven't shown any data. It almost seems like obvious. Right? So you can imagine that these neurons in the body is probably under some kind of tension. If the tension is too low, then they would shrink to build up the tension. If the tension is too big, then it grows to relax the tension. Because if it grows, if I stretch the rubber band, and if someone adds a little bit more rubber band, then it would relax the tension. Because it is more rubber band to be stretched. Right? If I hold the rubber band too taut, if I add a little bit to it, then the tension will go down. So this could be the growth mechanism. Okay, but is it just that? I will now show some examples. So what happens is this tension plays a significant role. So what we did is we took fruit flies and we cut these axons in the middle, and all we were doing is studying how is the vesicle density in the axon as well as at the end. So if you cut off the vesicle, cut off the axon in the middle, it turns out that vesicle at the end, which is supposed to be very high at the neuromuscular junction, as I showed you in the earlier view graph, is now dropped. So you can imagine, well, because there is no connection to the cell body, so they are not clustering anymore. Okay? So then we pulled this cut axon, and we find they have gone up again. Okay? So it needed the tension to cluster the vesicles. This has nothing to do with the growth. All we are saying is clustering of the vesicle at the end, which is essential for neurotransmission. Right? You can have action potentials, but if it goes all the way to find the soldiers are not there, there's nothing to fire. So clustering is essential. So this guy needed some kind of tension to keep the clustering going. Okay? So is it true that this was under really tension? This was a cut axon pulled by a micropipette, and this is the actual data. We can go through later on, but it's not necessary. So we needed to find out that are these axons under tension? And so we needed to find out some means to measure the tensions. I will skip that part. This is the engineering part, which is relatively simple. You need a spring to measure the tension. So if someone gives you a guitar string and say, what is the tension? So what would you do? You just hang a weight and see how much does the weight get it down, right? If you have a measure of the weight, then you can find the tension. So that's what we are doing here. So by doing that, we find that if you stretch an axon with the embryo living, this is one of the axons. This is about 80 microns in length, 300 nanometers in diameter. We are having a probe that is pulling the axon and giving rise to this force strain data, and it does not intercept at the origin, which means this axon was under tension to start with. Okay? And if you do many, many experiments, you see the tension is about 5 nanonewtons. Okay? So, very quickly, what I have shown is these axons are under tension in vivo. And if that tension is not there, then the vesicles at the end cannot be clustered. Okay? If you increase the tension, then vesicle density can go up. If I increase the tension by pulling it more, then you can increase the vesicle density by 3 times by just having about 10% of the stretch.
That's after how much time?
About after 30 minutes. Good point. So you stretch, hold the stretch for 30 minutes, vesicle density begins to build up more and more and more, and after 30 minutes you see they have gone up by 3 times. Yes?
So we have like focal adhesion of the cells, so those are anchor points where the cells actually adhere to.
Mm-hmm.
So in this case, does the axon also have like focal adhesion?
Yes, so when I showed the neuromuscular junction, so that's what the focal adhesion sites are. You also have integrins, you also have EK adhering junctions at the neuromuscular junction.
How dynamics are these structures?
So these structures are very complicated. So they are not like flat. So they are like interdigitated. So there's a large surface area, and that's where these integrins and other transmembrane proteins are involved. And they are dynamic because the studies have been shown that you can change the functionality of the integrins, and you can change the sequence of action potentials and the release of the vesicles as well.
So by creating tension, are you creating more stable microtubules that will help motor proteins to carry more neurotransmitters, or is it some other mechanism?
This increase part of it, we don't know. We don't know. I think that's a big question that we have to address. We have some hypothesis and some evidence of this hypothesis that one possibility is having more microtubules, another possibility is having more calcium getting in through the other stretch-activated ion channels that allow more ATP-driven motors to bring more vesicles at the end. But we have not pinned down the question yet.
What is the time frame? Because if microtubules, it will be like longer.
Correct. So this is a longer time frame. That was the question earlier asked. It takes about half an hour for this building process to complete, to saturate, which means it needs some signaling to happen, which means if it is microtubule polymerization, it needs longer time. So this might be one way that it carries more vesicles at the end.
Can we go back and forth if you reduce the
These experiments are such that they cannot be reversed, because once you dissect an embryo, you have only 2 hours to consider it living. So by the time you stretch the axon, you spend almost more than half an hour. You need another half an hour for the stretch to be held. So if you unload, by the time you unload or unstretch, we are crossing this 2-hour limit. So it would not be acceptable. So these are fairly challenging, although I'm getting it very fast. But we have not done the unloading experiment. Although we have not done unloading after loading experiment, we have done unloading before loading experiment, which you can do. because you have the axons coming all the way up to here, you can push the axon from the muscle side. Okay? So initially they are under tension, and then you unload it by bringing the end closer. Right? So that will cause the unloading. What do you think should happen? Based on what we just said. What's that?
The density will reduce.
About half an hour to destabilize the clustering. But beginning to build up the tension happens in about 9-10 minutes. So we are competing to create the unloading state against the shrinking ability. So in an experiment where you could unload and see what happens to the vesicles is not possible. The only way you can unload is by cutting it off, which we did. Vesicle density goes down. Right?
So is there myosin?
That's one of our hypotheses. The myosin is there to create this tension, actomyosin machinery.
So you can deactivate myosin.
It's because these neurons have myelin sheaths and many other protective mechanisms. These experiments are, and these are all in vivo. It's not guaranteed that you can easily deactivate them. And also there are many different myosins. We don't know which one is causing the tension, but myosin playing a role in creating the tension is one of the hypotheses, certainly. Okay, I'll skip this one. So the fact that neurons might be under tension has been thought about by neuroscientists for some time. One of the earlier paper, well, I think one of the first paper that came out was 1997 by Van Essen, and he made a proposition that the brain neurons have this tension, which gives rise to this folded structure. It's like buckling a sponge, and this also it creates much more optimum circuitry. Has never been proven experimentally, as far as I know, in adult brain. But this was a proposition that it's the tension in the neurons that give rise to this highly organized structure and organized and efficient circuitry. Okay? So if you think of a buckling process, if the buckling column is very small, you cannot buckle. Right? You end up having just tension or compression but not buckling. If the ruler is very long, if you push a little bit, it buckles. Right? So Van Essen argued that when the brain size is below a certain size, then you should not see this apical folding. And then he shows that in the whole spectrum of the animal size, brains which are below a certain size does not have this apical folding. Above a certain size, they see the apical folding, which indicates that the tension is probably everywhere in all size scales. Okay, so I think don't put too much attention on this data, but I think I already mentioned that the way this tension plays a role is when you stretch, the axons grow, and by growing, it can relax the tension. And we have done the experiments to show that it does relax the tension. And if it indeed grows, that means if it adds more cytoskeleton, if it adds more microtubules, then what would you expect? In an experiment where you stretch an axon, you hold the stretch for some time, and then you think it is growing by bringing more cytoskeleton, by bringing more content, by becoming fatter maybe even. What would you expect mechanically? So you take a rope, you stretch the rope, and then you double the size of the rope. Then what do you expect? Increase in stiffness, right? Because it's like more material is there to stretch. That's exactly what happens. So if you just think of the stiffness of the axon, and if you hold the tension for some time, the stiffness doubles in many of the axons. It depends on how much you stretched in the first place, but if you stretch in a physiological range, then the stiffness doubles in about half an hour. So the only way this can happen, without any other biological interpretation, is more material has to be there. Or it could have by realignment, but there is not much room to realign because they were only about 300 nanometers to start with. So the growth has to happen by the material has to be added. Now, if you think about what we discussed earlier, these are highly rich structures, these axons. They have ion channels, microtubules, vesicles, highly dynamic. So when it grows in half an hour, nobody has shown that yet, but I would imagine that it's not just growing just by putting microtubules, but it's probably adding all the ion channels, all the motor proteins that are needed to sustain this structure. So that is essentially what happens as we develop, that the force applied by the developing organs, the axons or neurons are responding to it, essentially the axons, and they're increasing their diameter because that's what they need for more conduction. And eventually, when the development ends, no more growth happens, it somewhat maintains a steady tension, and it doesn't overgrow anymore. If the tension happens to be too low, then it would build the tension by shrinking and stabilize it. And again, we have shown that maybe here in one graph that the tension builds up if you let it relax and hold the displacements. Okay, so I know my time is up, but I would like to end by saying that just one notion. I'll take maybe 2 minutes. 2 minutes. Okay. This is a very open-ended thought connecting to the mechanics. Is like if you look at the stadium, we are all human beings, and each one is independently different than the other ones, right? Individuality is essentially what makes us so unique, and this individuality comes from the way we were developed. We were cultures, our languages, the societies, and all those things that is happening. So people have thought about this for over 200 years, and at least for the last 1 century, the question came that what gives rise to our individuality? What makes us different from one another? Everything has to be happening in the brain, right? That's what makes us think. And everything happens in these synapses, many, many synapses that we have. And I also briefly mentioned that the synaptic strength depends on the type of experience that each synapse undergoes through. The synaptic plasticity changes. So is it possible that if you think of these many, many neurons in the brain, that as we develop, as we go through our life experiences, we also have different tensions in different neurons. And eventually, if we have a tension map, just like sometimes we think of having synaptic plasticity map, where we look at the number of vesicles clustered at the end. If we had a tension map in the brain where different axons have different tensions based on different experiences we have, could we decipher our essence of individuality? So if you think of role of mechanics, that's where it comes from. That as we grow, the neurons are being stretched. Neurons have their inherent ability to shrink, to build up that tension. And this tension seems to be related to the growth as well as clustering of the vesicles at the end. And it is that clustering of vesicles that is essential for action potential to act on them, release the vesicles, and transmit the information. So I'll stop here, and if you have any more questions, we can have some addressed after the second lecture.